Hello students, welcome back to my channel. In this video, I am going to give you a brief lecture on nucleic acids. So there are two nucleic acids, they are nothing but DNA and RNA. DNA is nothing but deoxyribonucleic acid, RNA is nothing but ribonucleic acid. Okay, so they are the genetic materials. They consist all the genetic information which is necessary for the protein synthesis and also for heredity. They consist all the genetic information. So nucleic acids are the complex informational biomolecules. They are present in all living organisms and also viruses. So they were first discovered and isolated from pus cells. They are obtained from discarded hospital bandages by a scientist called Friedrich Miescher. So he discovered nucleic acids for the first time in the pus cells, obtained from the discarded hospital bandages. So he thought that they were phosphorus-rich nuclear protein molecules. The term nucleic acid is given by a scientist Altman. In the year 1889, he gave the term nucleic acid. DNA is a genetic material that is found in all cells. Every cell has its own DNA content. So that DNA is present in the nucleus. Okay. So every cell consists the genetic material that is DNA enclosed in the nucleus. Okay. But it is absent in plant viruses. So in case of plant viruses, there the RNA is the genetic material. There the DNA is absent. So let's see the components of nucleic acids. In DNA or RNA, there are three components. They are pentose sugar, second one is phosphate group, and third one is nitrogen bases. So these are the three components that are common for DNA and RNA. So let's see one by one. So let's see the first component, that is nothing but pentose sugar. So the scientist Levine, he identified the ribose sugar is the main constituent of nucleic acids. So the ribose sugar is the main constituent of nucleic acid structure. In case of RNA, ribose sugar is present. But in the case of DNA, deoxyribose sugar is present. So let's see the structure of ribose sugar and deoxyribose sugar, and let's see the differences between ribose sugar and deoxyribose sugar. So in case of RNA, ribose sugar is present. In the case of DNA, deoxyribose sugar is present. So this is the deoxyribose sugar and this is ribose sugar. The difference is one oxygen atom present at the second carbon of ribose sugar is missing in the deoxyribose sugar. So that is only the difference between ribose sugar and deoxyribose sugar. So RNA consists ribose sugar whereas DNA consists deoxyribose sugar. So the carbon atoms of ribose sugar are denoted as First carbon, second carbon, third carbon, fourth carbon and fifth carbon. So this is a pentose sugar. Okay. So let's see the next second component, that is nothing but phosphate group. So if you see the nucleic acid structure, there you will find the phosphate groups, they alternate with the pentose sugars. Okay. Let's see the phosphoric acid structure. It consists 3 active hydroxyl groups. So they involved in the phosphodiester bond. So they alternates with the sugar molecules, pentose sugar molecules. So let's see, actually this phosphate group is present between 2 pentose sugars. Let's see how it will involved in the phosphodiester bond formation. So let's see the structure. This is the phosphodiester bond formed in between 2 pentose sugars. So this is the phosphoric acid group present in between 2 pentose sugars. So okay, each phosphate group is joined to the third carbon of one pentose sugar to the fifth carbon of another pentose sugar. In this way, it is results in the formation of phosphodiester bond. So this is called phosphodiester bond. So let's see the bond. 5 prime carbon, oxygen, and then phosphate, and then again oxygen, and then again 3 prime carbon. So this is the phosphodiester bond. So 5 prime carbon, oxygen, phosphate, again oxygen, so 3 prime carbon. Okay, let's see the third component of nucleic acid structure, that is nothing but nitrogen bases. So nitrogen bases of nucleic acids are of 2 types. First one is purines. Second one is pyrimidines. So purines are nothing but adenine and guanine. Are called as purines. It is designated as A. It is denoted as G. So pyrimidines are nothing but cytosine and thymine. So it is designated as C and it is designated as T. So in the case of RNA, so purines are common for both DNA and RNA, whereas in the case of pyrimidines, cytosine is common for DNA and RNA, but instead of thymine, RNA consists another pyrimidine, that is nothing but uracil, which is designated as U. So in case of RNA, instead of thymine, there is a pyrimidine uracil is present. Okay. So let's see the detailed structure of purines first. So these are the structures of purines. So this is the structure of adenine and this is the structure of guanine. Purines consist 2 rings. One ring is 6 carbon ring. So that is similar to benzene ring. So this ring is called as pyrimidine ring. 6 carbons, pyrimidine ring is present. And also they consist 5 carbon ring, that is nothing but imidazole ring. So pyrimidine ring is just like benzene ring, but the difference is, so first and third carbons, they are occupied by nitrogen instead of carbon. Though that is the difference. So here also you can see the same. Okay. So, but in the case of imidazole ring, that is similar to uric acid, but the difference is 7th and ninth positions. So, they were occupied by nitrogen instead of carbon. So, that is the difference. Okay. So, if you see the difference between adenine and guanine, in case of adenine, in the 6th position, there is an amino group. Whereas in the case of guanine, in the 6th position, there is a keto group. Keto group is nothing but C double bond O group. Okay. So, here it is amino group. But in the case of guanine, it is keto group. So, but in addition to keto group, the guanine consists an additional NH2 group, that is nothing but amino group at position 2. Okay. So, the ninth position nitrogen covalently bind to the first carbon of pentose sugar. So, the ninth position nitrogen is covalently bind to the first carbon of pentose sugar. In this way, they attached to the ribose sugar in the first position. So, the bond established in between these two is called as N-glycosidic bond. The bond established between the sugar and nitrogen base is nothing but N-glycosidic bond. I will explain this later. So, let's see the second type of nitrogen base is nothing but pyrimidines. Okay. So, cytosine and thymine are called as pyrimidines. They are present in DNA, whereas in the case of RNA, thymine is replaced by uracil. Okay. So, you can see the structures of pyrimidine bases. So, all pyrimidine bases consist. So, the pyrimidine rings. Pyrimidine ring is nothing but which is just like a benzene ring, but the first and third carbons of a benzene ring were replaced by nitrogen. That ring is called as pyrimidine ring, which is common for pyrimidines. Pyrimidines are having pyrimidine ring. There the structure consists first and third carbons occupied by nitrogen instead of carbon. So, that is the difference between benzene ring and pyrimidine ring. So let's see first the common feature for all the pyrimidine bases. So, all three pyrimidine bases consist of second position. In their second position, they consist a keto group. That is C double bond O group. Okay, that is common for three pyrimidines. But the difference is, so cytosine consists amino group at the fourth position, whereas thymine consists methyl group at the fifth position, and also fourth position is consisting keto group. So, and uracil consisting keto group at the fourth position. So, uracil consisting keto group at the fourth position. Only difference between uracil and thymine is fifth carbon consisting methyl group. That is only the difference between uracil and thymine. Okay, that's why thymine is also called as a 5-methyl uracil. Okay, thymine is also called as 5-methyl uracil. Pyrimidines attached to the pentose sugar at the first position, whereas purines attached to the first carbon of pentose sugar and ninth position of imidazole ring. But here they don't have imidazole ring, so they are attached to the pentose sugar at the first position. Okay, so you should remember. So let's see the next important structure of the nucleic acids is nothing but nucleosides. So nitrogen base plus sugar is called as nucleosides. So nitrogen bases, either it can be purine or it can be pyrimidine. purine or pyrimidine, they are linked to the pentose sugar with a bond called as N-glycosidic bond. So the bond present in between base and sugar is nothing but N-glycosidic bond. Okay. So the glycosidic linkages established in between the ninth position nitrogen of purines is bind to first carbon of pentose sugar. Okay, so that's why these are called as N9 glycosides. Whereas the first position of pyrimidine ring, they don't have imidazole ring. Okay, so ninth position of imidazole ring of purine is bind to the first carbon of pentose sugar. Whereas first position of nitrogen, first position is occupied by nitrogen, so that nitrogen bind to the first carbon of pentose sugar. So that's why pyrimidines are called as N1 glycosides. N1 glycosides. Okay, so the purine nucleosides are called as N9 glycosides, whereas pyrimidine nucleosides are called as N1 glycosides. If the nucleoside consists ribose sugar, then the nucleosides are called as ribonucleosides. Present in RNA. So if the sugar is deoxyribose sugar, then the nucleosides consist deoxyribonucleosides. So then the nucleosides are called as deoxyribonucleosides. They are present in DNA. So the bases, they can be purines or pyrimidines. So let's see the ribonucleosides first. If the base is adenine plus ribose sugar, that nucleoside is called as adenosine. So if the base is guanine, the sugar is ribose sugar, then it is called as guanosine. If the base is cytosine, the sugar is ribose sugar, that nucleoside is called as cytidine. If the base is uracil, it is RNA, you should remember this. In case of RNA, uracil is present instead of thymine. Plus ribose sugar, that is called as uridine. So these are the 4 ribonucleosides present in RNA. So let's see the 4 deoxyribonucleosides. Okay. So if the adenine is present plus deoxyribose sugar is present in the nucleoside, then it is called as deoxyadenosine. So if guanine is present plus deoxyribose sugar is present, then it is called as deoxyguanosine. If cytosine is present with deoxyribose sugar, that is termed as deoxycytidine. If thymine is present with deoxyribose sugar, that is termed as deoxythymidine. So here you have to remember one thing. So the purine nucleosides are end with the suffix "sine" or "sine", whereas the pyrimidine nucleosides they end with the "dine". They end with the suffix "dine". So purine nucleosides, they end with the suffix "sine", whereas pyrimidine nucleosides, they end with the suffix "dine". You have to remember this. Okay. So this is the nucleoside structure. So these two are called as nucleoside. Base plus sugar. So that is called as nucleoside. So this is pentose sugar and this is the base. So base is attached to the sugar at first position, you should remember. So at the fifth position, it consists a phosphoric acid group. Okay, a phosphate group. Okay, totally this is called as phosphate plus sugar plus base, that is equal to nucleotide. Okay, so that is equal to nucleotide. So now we are going to see what is nucleotide in detail. Nucleotide is nothing but phosphate, sugar and base. So the phosphate group is attached to the nucleoside, then it is called as nucleotide. Okay. So the phosphate group attached to the fifth position of ribose sugar. It can be ribose sugar or it can be deoxyribose sugar. So the phosphate group is attached to the fifth position of the sugar. If it is ribose sugar, then the nucleotides are called as ribonucleotides. If it is a deoxyribose sugar, the nucleotides are called as deoxyribonucleotides. Let's see 4 ribonucleotides first and then 4 deoxyribonucleotides later. So here you have to remember one thing. So the bond present in between base and sugar is called as N-glycosidic bond, whereas the bond present in between two sugars, here another sugar is attached here. Okay, so the bond present in between two sugars is nothing but phosphodiester bond, which is a backbone for DNA structure. So the bond present between sugar and base is called as N-glycosidic bond. Okay. Let's see first 4 ribonucleotides. So let's see the first 4 ribonucleotides. So they are 5' adenylate acid, 5' guanylic acid, 5' cytidyllic acid, 5' thymidyllic acid. Let's see the 4 deoxyribonucleotides. That means it consists deoxyribose sugar. That's why they are called as deoxyribonucleotides. Okay. First one is, if adenine is present, that is called deoxyadenylic acid. The guanine is present, so then that is called as deoxyguanylic acid. So if the cytosine is present, then it is called deoxycytosine. cydidylic acid. If the thymine is present, here it is the uracil, then it is called as uridylic acid. Okay. So if thymine is present, that is called deoxythymidyllic acid. So in case of RNA, uracil is present instead of thymine. You should. So in DNA, these deoxyribotides are deoxyribonucleotides. They joined one another to form a linear DNA strand. Okay. So the backbone of DNA is nothing but phosphodiester bond, which is established in between two sugar molecules. So let's see the detailed structure. Here you can see the detailed structure of DNA. So actually this is one strand and this is another strand. That is one strand and this is another strand. Here you can see the phosphodiester backbone of DNA. So this is the one strand and this is the another strand. They joined together by a hydrogen bonds. They are established between two bases. Okay, in between C and G there are 3 hydrogen bonds. In between thymine and adenine there are 2 hydrogen bonds. Okay, so I will explain this later. So let's see the structure of one strand. So here you can see. So in between two sugars, one phosphate group is present. So this is called phosphodiester bond is established between the two sugars. So it is attached in between the 5th carbon of one sugar and the 3rd carbon of another sugar. You have to remember this. That is a phosphodiester bond established in between the two sugar molecules. Okay. So in case of these two are called as nucleoside, as I told you earlier. These two are the combination of sugar and base is called as nucleoside. So the sugar is linked to the either purine or pyrimidine bases. They are nothing but nitrogen bases with the bond is nothing but N-glycosidic bond. Okay. So totally this is this is called as nucleotide. Okay, nucleotide. The combination of phosphate, sugar and base is called as nucleotide. Base plus sugar is called as nucleoside. Okay, so this is the one strand attached to the another strand with the help of hydrogen bonds. I will explain this in the next session. Okay, so let's see the primary structure of DNA. So this is the primary structure of DNA, students. So you can see the DNA molecule. So it is a combination of two parallel strands. Okay, two linear strands, they run opposite to each other. So they are anti-parallel to each other. So if you see the single strand, it is a polymer of nucleotides. That means it consists many nucleotides. They join together by a phosphodiester bond. So the backbone of DNA is phosphodiester bond established between the sugar molecules. So here you can see the priming, 3' and 5'. So they are opposite to each other. At this end, the sugar consists 3' OH group free. There is no phosphate group. Okay. So the free OH group is represented as 3' end. So here you can see the another priming, that is 5' end. Here there is no phosphate group. That's why here the position of sugar there is a free OH group. So that's why here I am representing as 5'. So that is the end part of the DNA. So they are showing polarity in this way. Okay, they are anti-parallel to each other. You can see the nucleotide. This is called nucleotide. Okay, so nucleotide is nothing but phosphate, sugar plus base. So in between base and sugar there is an N-glycosidic bond. Between two sugars there is a phosphodiester bond. Totally this is called nucleotide. So DNA is a polymer of nucleotides. Okay. So this is the one strand. This is having another parallel strand. There you will find another nitrogen base, that is thymine, which is attached to the adenine with the two hydrogen bonds. Here you can see the another hydrogen base, that is cytosine. So that is attached to the guanine with the three hydrogen bonds. So in this way you can see. So here also another polynucleotide chain attaches to the another strand with the help of hydrogen bonds. So the bonds in between two strands of DNA are nothing but hydrogen bonds. You have to remember this. So the two strands of DNA are when they come together, adenine is always binds with the thymine, whereas guanine always binds with the cytosine, vice versa. This is the complementary nature of DNA. In this way, the two strands are bind together. Okay students, this is about the nucleic acids. Let's see the detailed structure and Watson and Crick model of DNA in the next video. Thank you students.